Okay, so thank you, Randy, and the organizing committee for giving me this opportunity to share our failures. So basically, I've been working on imaging vesicles for about 15 years, and I'm quite humbled to be here to teach you, quote unquote, but rather I just want to share what worked and what had not worked for us. So let's begin that with, let me see if I can go to the next one, Canadian bacon. So what do you have in your mind when you hear about Canadian bacon? If you're not from, is anyone from Canada here? We have a few. Good. Only like less than 1%. All right. So do you know what a Canadian bacon is? Female? Oh, you actually know that. So that's what I meant. Like Canadian bacon usually in people's mind, especially with my American friends, like they usually make fun of me. It's like, Charles, where's your Canadian bacon? So turned out to be a ham. So vesicle imaging is pretty much like this, that a lot of people say seeing is believing. That only happens when it's well planned. And I think to start with Rink's talk this morning is like, who was actually born before and after 1988? And I was just thinking, I'm definitely the before 1988 group. And I kept thinking, if I were you who's very young and just started your career and then entered Vesicle, I think this morning's session has been very intimidating for some of you. So I try to make this talk as lighthearted as possible while touching on the main principles without overwhelming you. But let me just be really clear that there is no one perfect method for vesicle imaging or labeling. Just keep that in mind. Everything has its pros and cons. And each method requires characterization and or validation. And this is like probably the take-home point for the whole entire talk. Now, why do you want to do vesicle imaging? And usually people like to do imaging when you see that the vesicles carry a biological role. And in fact, they are good because it allows you to simultaneously monitor spatial temporal resolution and hopefully observe the morphological and physiological changes that you treat your cells or systems with. Not only that, if you can do your imaging well enough, potentially you're not only looking at a single cell, you can look at them at cellular level, tissue level, organ level, or whole organism as a whole. And even better, if you do it right, you can do non- or minimal invasive imaging of the vesicles. Now, further to that is, can you imagine trying to quantify the vesicles in vitro and in vivo? This is a key point, but it's really hard to achieve. But at least we dare to say that with the technologies we have now, we can at least semi-quantify vesicles. And other things for doing vesicle imaging is that you can avoid sample processing artifacts. And not only that, if you can avoid specimen modification or degradation during the process, that's what imaging is really good for. Sampling error is also something that happens if you're collecting clinical samples. So again, if you can do molecular imaging, it really helps you with your research. But usually I would say that, I'm trying to make this talk a little bit more philosophical, is that before you begin your research, please think deeply about what you're trying to answer. Like, what is your proposed question? Rather than pursuing pretty images. Okay, I kept saying this. Don't pursue pretty images. I can give you a lot of pretty images, which really doesn't mean much, right? So what is your proposed vesicle question? Is it related to development, diseases, cancer, neurological disease, whatnot? Or are you trying to make devices? Okay, biomarkers. Okay, would vesicle imaging help you? Therapy, are you trying to develop therapeutic vesicles? Think about out of these questions really deeply before you approach imaging, because it will answer the following question: what is the sensitivity that's required for your imaging, and what is the resolution that would best fit to answer your questions? Now, Randy told me not to put in much about my work, but in my former life, these are probably the prettiest images I can show you. When I was in Dr. Zandra Bricksfield's lab, that I was able to track vesicles by labeling 2 cells, and then not only that, we can also do in vivo imaging. So to go into further, I would emphasize on in vivo studies. I think a lot of people use different organisms and model to do their study, and this is something to really think about when you're trying to label and visualize your system, because they are with different sizes, and if you're trying to see the vesicle biological activity inside these model organisms, you really have to think about how you wish to label them and how you wish to image them. Now, after you've identified your biological questions, then you have to think about specificity. So can you label your vesicles specifically? Does it yield accurate spatial as well as temporal resolution? and can it yield resolution that you want? Do you need whole animal resolution, or do you need super resolution such that you can observe them subcellularly? Does it provide sufficient signal-to-noise ratio? So these are the things that you really have to think about. If you're still with me, and if you can answer all these questions when you're planning your experiment, you're on the right track. Okay, don't pursue just pretty images, because seriously, it's very, very misleading. Now, just to go over really quickly about ways to image vesicles, if you just based on their biophysical property, you can image them by dynamic light scattering, NTAs that I've utilized on. By microscopy, there's EM, SEM, TEM, or you can do AFM. Optically, you can do a lot of stuff. This is not a microscopy workshop, so I won't go into details, but if you're interested, there's a ton to go through. You can also rely on chemiluminescence or bioluminescence to image your vesicles. For medical imaging, you can do MRI, SPECT, CT, and the bottom ones are more categorized into the molecular imaging category. Now, how do you label your vesicles? You can do them by antibody, or you can do them by reporter gene. Usually this would be more EV protein specific labeling that you can do. Or you can do general labeling. You can do lipophilic dye. You can do clay chemistry, so you can metabolically label the membrane and then do clay chemistry to label vesicles in general. Again, you can also have general labeling reporters. And you can also have radionucleotides or other contrast agent to help you label. Now, if you want to see them live, I categorize them based on the labeling location. You can label the vesicles within. You can label them on the transmembrane, the inner membrane, or the outer membrane. Those are all feasible. But you have to think about the pros and cons. If you label it inside, would it affect the cargo content? If you label on the outside, would it actually yield spherical hindrance? Would it actually prevent binding between proteins and other stuff? Because we now know that there is corona, and there are also glycocalyx and all the stuff on the outside. So if you label on the outside, would it affect the membrane dynamics? And also, just like what David was mentioning, would it affect organotropism? So those are things that you need to really think about and how it relates to your labeling strategy. Now, to go into antibody-based labeling, they're usually very specific on the marker that they express. So that's a pro if you're into, for example, like CD63, CD81, okay, but pros, and that's also the con, because you're only looking at a very specific population of vesicles. And the labeling procedure has to be really exact, such that you don't have like false positive binding and stuff like that. And as I'll mention again, you need a lot of protocol controls to make sure that what you're seeing is not false positives. Now, these are just some of the samples I mentioned. Now, in terms of reporter-based labeling, you can have general labeling or specific vesicle population, and they typically do not require the vesicle to be stained by, say, lipophilic dyes. Now, the cons to that is that if you're not familiar with molecular cloning or, say, lentivirus or transfection experiments, this may be a little bit troublesome for you. Not only that, you typically require the donor cell for your vesicles to express the reporter. Again, this may be a little bit limiting to some, especially if you're working with primary cells. And again, it does require control to make sure that you're seeing true signal. Now, for the radionucleotide or contrast agent-based labeling, they have a really good advantage in terms of their tissue depth. Yeah. But for the cons to that is not a lot of people have access to these contrast agent, and they could be very expensive to work with. At the same time, they require control, and some examples are listed right here, which I won't go into, but you can check those out. Okay. So for dye-based labeling, this is something that I kind of want to emphasize on, and I know that a lot of people really like to use dye, is that they really are great in providing you an awesome signal, and they are relatively easy to apply. The cons to that is you have to be really careful about the labeling procedure, and at the same time, you do need to implement control to make sure that you are really looking at label vesicles. And some examples to this is the lipophilic dyes, including PKH, Cellview, dye. I know there are new dyes out there. There are also other dyes such as CalCAM or CFSC, and I think there are a couple good papers comparing how the different labeling dyes work. I think there's one paper that suggests that CFSC is one of the better one. So go read the paper and decide for yourself. But I just want to mention this, is when you are labeling your vesicle, you should think about the specificity, the dye's half-life when compared to the vesicle's half-life, as well as efficiency in labeling. So one example is PKH. I think when I started doing vesicle work, PKH was like, the dye to go to. That's like back in 2010-ish. Yeah, I'm old. And then, but what is PKH? PKH is essentially like aliphatic dye with really long hydrocarbon chain. So the longer it is, the longer the half-life it is. And they are great when labeling immune cells. You can track them for a month. And this is actually the half-life of those dyes in vivo. This is not in vitro. They can last up to 100 days. So think about it. You label your vesicles. Do you expect them to last for 100 days? Chances are not. Yeah. But a lot of people use it. Later, a lot of people start to report that these are lipophilic dyes. They actually do form nano-sized micelles, which can give you false positive signals. Once you label them, just like the Easter egg on the right, if you know what this is, and then once you label them, you have to get rid of the dyes, right? How do you get rid of the excess dye? So that's another question. And then you learn from Rosella's talk this morning, the isolation itself is complicated enough. Imagine trying to get rid of the dyes. Problematic. Similar principle applies to other lipophilic dyes. I'm not saying that there are no good dyes. It's just that you need to know where you're working with. Yeah. So the dyes are not evil. Rather, the user should know what's going on. One thing that not many people look into is the labeling efficiency of the dyes. How do you know if you label 100% of your vesicles? I don't think anyone dare to claim that. I definitely do not dare to claim that. But keep that in mind when you're applying the different labeling methods, as well as its half-life. Now, how does that relate to translation? Imaging is almost critical with preclinical research. Because imagine, I think a lot of people are trying to tell you that vesicle is effective in regenerative medicine, or you're trying to see if a vesicle has a disease-promoting effect. So where are they going about in your body? What is the dosage that's relevant to your therapeutic vesicles? Those things are largely unanswered. I think to a lot of us here, we usually try to administer as much vesicle as possible until you see a phenotype. Our recent studies show that if you administer too much, it could kill your animal. This is very donor cell dependent. But if you administer too much, it may not be physiologically relevant. And for preclinical research, it's important to establish the dosage, the half-life, so that they can assess the pharmacokinetics of the vesicles. And conversely, if you apply this to the disease model you're studying with, it will kind of make your study a bit more physiologically relevant, rather than just overexpressing, overdosing until you see an effect. So just to keep things in mind, I'm going to keep this simple. Just report as much detail as you can for your vesicle labeling methods, as well as how you image it. I mean, just tell us about your parameters and stuff like that. I think that's important. I mean, the purpose of this is not to intimidate you, but rather for other people to repeat your work. And similar stuff apply to quantification. I mean, tell us the threshold that you set and what program did you use so that we can repeat the work. And again, apply controls. It sounds a little bit vague, but just keep thinking, how do I prove to my peers that what you're seeing is real? Okay, and etc. Now, I'm keeping this very simple. What is on the horizon? I don't know if you're too young or too old to know Daft Punk. Oh God, all the laughing ones are seniors like me. But I really like this because on the top left, have you heard of the song Harder, Better, Faster, Stronger? You did? You gotta read the Japanese cover art. It says that shikoto wa waranai, and I kind of check Google Translate. It says that work never ends. That's rather practical, but again, a lot of people are trying to develop better reporters to be more specific, to yield higher signal-to-noise ratio. Great, but just keep those principles in mind about what I mentioned: the specificity, the half-life, the efficiency. How does it relate to the question that you're trying to answer? Better dye. A lot of companies, including our sponsors here, they report that they have better dyes. I'm not saying anyone is better than the other, but give it a try with all the principles that I mentioned. Are they specific? Half-life, blah, blah, blah. Keep those in mind. Better instruments? Oh, there are so many things that we can go into. You have light sheet microscopy, expansion microscopy, CLAM, CLEM. There's also CLAM, like CLAM. Coated array light, I forgot. Anyway, so there are a lot of new microscopy coming out that is just going on, but see which one fits better for your work. And also something that I didn't mention as much is are there non-labeling method? There are a lot actually, like Raman spectroscopy, FTIR, third harmonic generation. I think this is especially from Stephen Bopar's lab from Champaign. Yeah, so they're doing a lot of fantastic work. But same question applies to you. Do you really need that level of resolution to answer your question, right? better image is not necessarily better. So I'm trying to keep this talk quite philosophical but logical so that you catch the principle for imaging. And I hope this has been helpful to you. Thank you.